Okay, cofactors are non-protein enzyme helpers. Cofactors may be inorganic, such as metals, ions, magnesium is a cofactor. Manganese is a cofactor sometimes. Calcium sometimes is a cofactor. An organic factor, so organic ones are coenzymes. Coenzymes are organic ones like vitamins. Vitamins are coenzymes, and cofactors are like, as I said, they are like minerals. Okay, so they are inorganic. They are not organic and organic. You know the difference between organic and not organic. Coenzymes include vitamins. Yes, they said that here. Okay, what are enzymes inhibitors and their examples? Competitive inhibitors bind to active site of an enzyme competing with the substrate. So if beside sucrose that attaches to sucrase right here, something else attaches here, then sucrose cannot break down. And that's what competitive inhibitor means. Non-competitive inhibitors bind to another part of the enzyme, causing the enzyme to change shape and make active sites. So if you have an enzyme like this, okay, and this is active site right here, and competitive inhibitor attaches to here. Am I making some sense? Not sucrose. This, let's say this is sucrase. This enzyme is sucrase. So not sucrose, something else. I'm making it up. Like galactose. Galactose attaches here. I'm making it up. Okay. Then this enzyme cannot work. And sucrose comes here. It cannot be broken down to fructose and glucose. But the other one, the non-competitive inhibitor, will attach right here. And when it attaches here, what happens to the shape of the enzyme? The enzyme becomes like this. The enzyme becomes like this, and nothing can attach to it now. The shape of the enzyme changes. And that's what the non-competitive inhibitor is. Examples of inhibitors include toxins, poisons, pesticides, and antibiotics. Antibiotics, of course, for bacteria, not for us. Okay, so here they are. Normal binding, this would be your substrate, such as sucrose. This is sucrase. And then, of course, when something competitive inhibitors attaches here, then this sucrose cannot attach here. That would be a competitive inhibitor. A non-competitive inhibitor, as I draw it on the board, attaches right here, not on the active side. The shape changes. This is not a perfect lock and key. Am I making some sense? Lock and key, we talked about this. This is not a perfect fit. It will not sucrose will not break down to glucose and fructose. That's what I'm talking about. Okay, allosteric regulation may either inhibit or stimulate an enzyme activity, and I guess there is some pictures coming show you that. Allosteric regulations occur when regulatory molecules bind to protein at one site and affect the protein's functions and another site. Oh, no pictures. Okay. There is more about allosteric activation. Of inhibition continues. Most allosterically regulated enzymes are made from polypeptide subunits. Each enzyme has active and inactive forms. Of course, you know this statement. Okay, the binding of an activator stabilizes the active form of the enzyme. The binding of inhibitor stabilizes the inactive form of the enzyme, right here. Okay, so this is an enzyme. They're showing you it has 4 subunits, 1, 2, 3, 4. It's an enzyme, allosteric enzyme with 4 subunits, 1, 2, 3, 4 subunits. Active sites, these are the active sites. Okay, so the activator will come and attach to the enzyme, and then what stabilizes forms. Now what happens if you have cooperatively another type of allosteric enzyme, the substrate comes and attach here, then the enzyme will not be active. These are, if the enzyme changes its activity, non-functional active sites, and that's what the inhibitor is. The inhibitor comes here and attaches the shape. It changes the shape of the enzyme, so this is not active, but this form is the active form. And of course, cooperatively type, another type, this of course, this is the enzyme that is acting normally. Cooperativity of allosteric regulation that amplify enzyme activity. Yes, one subunit molecule primes an enzyme to act an additional substrate molecules more readily, and then cooperativity is allosteric because of all of these goodies, which I'm not too worried about it here on cooperativity. Feedback inhibition, what Reno was asking about, the last portion of this chapter, what is feedback inhibition? In feedback inhibition, the end product of metabolic pathway shut down the pathway. So the end product, sometimes when you have this enzyme, sometimes when you have sucrose, for example, well, he give you another example. Let's go by the text. I don't want to confuse you. Then glucose comes here and stops sucrase. You will see some examples. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing more products that it is needed. What is this example I gave you this morning? The light. If you leave the light here at night, you're wasting the light. Same thing here. So there is a mechanism to turn off the enzyme. There is a mechanism to turn off the DNA. Again, I hope I'm bringing DNA to a picture because of later on in the semester. But anyhow, here it is. So you have an enzyme, and then you have he gave you a good example, not my example of sucrose, 3-iodine in active site. What is 3-iodine? Do you remember that? Anybody? It's what? It's an amino acid. It's one of your amino acids that have what? Do you remember that? Alicia? No? It has sulfur. Remember? It has there are 2 amino acids that have sulfur, and 3-iodine is one of them. But anyhow, so you have this enzyme, and this amino acids goes in there and become intermediate A, another enzyme turn it to another intermediate, and to another intermediate, to another intermediate, eventually at the end becomes isoleucine. What is isoleucine? Do you remember that? Have you ever heard of isoleucine in your life before? It's what? Ariel, very good. It's what? It's an amino acid. Why are you bashful? You have the answer. Why are you being bashful? You're right. Okay, so it's an amino acid. One amino acid turns into another amino acids, but in between there is a pathway. Do I make sense? They call it metabolic pathway. There is a metabolic pathway. A becomes B, becomes C, becomes D, and eventually becomes isoleucine. Of course, there is an enzyme for each one of them. There is enzyme for each one of them. Then what will happen that isoleucine comes and sits right here. Isoleucine used up by the cell, and isoleucine sits on that enzyme right here. Do you guys see that? And then the shape of the enzyme changes. When the shape of the enzyme changes, it cannot take 3-iodine and turn it into isoleucine anymore. This is the regulation mechanism of an enzyme. Okay, this is all of the stuff that the feedback inhibition. That's it. This is a feedback inhibition. It inhibit that enzyme from making 3-iodine to turn into isoleucine. When there is too much of it inside of the cell. When there is too much of it, too much of isoleucine inside of the cell, then the enzyme needs to stop. Okay, I hope I'm making some sense. Localization on an enzyme within the cell. Again, I don't know, your textbook goes haywire sometimes. There are cells everywhere. He's saying, well, mitochondria have a lot of enzymes, which we're going to discuss but there are enzymes everywhere. Golgi apparatus has enzymes. Rough endoplasmic reticulum have enzymes. Nucleus have enzymes. So enzymes, the cytoplasm, inside of cytoplasm, there are lots of enzymes. So that's what your textbook is talking about. Eukaryotic cell, let's read it. Structure within the cell helps bring in order to metabolic pathways. Yes, some enzymes act as, this is mine, sorry. Okay, some enzymes act as the structural components of membrane. Of course, in eukaryotic cells, some enzymes resides in specific organelles. For example, enzymes in cellular respiration located in mitochondria. Okay, am I done? Yes, this is mitochondria. We are done with this chapter. Is there any question from this chapter? Let's go to chapter 9. Chapter 9 goes over cellular respiration. Right. Where does respiration occurs in you guys? Huh? Just regularly, somebody grabs your collar in the mall and say, where is respiration? What do you say? In what? You said it. In what? No, not even. Huh?
Lungs.
Lungs. Your lungs. Your lungs on the right side of your body has 3 lobes. The one on the left side has 2 lobes. Why is that? Huh? 3 lobes versus 2 lobes. Because you have your heart on the left side. So your left side has 2 lobes of lungs and the right side has 3 lobes. Am I making sense? That is different than what we are going to discuss in this chapter. That respiration that happens in your lungs is different than the respiration we are going to talk about. This is cellular respiration. Your lung respiration is just respiration. We call it respiration. You go to a doctor, I have bad respiration. And the doctor says, you mean cellular respiration? You say, no, doc. Where did you go to medical school? You ask the doctor. My cellular respiration is fine, but my respiration is not good. 2 different things. Am I making some sense? 2 different things. Do not get them confused. You go to a doctor and the doctor says your respiration is not good. It means your lungs are not functioning good. And then here in biology, you're learning about cellular respiration. Catherine, am I making some sense? Cellular. Of course, this is mitochondria, right? This is not a cell. This is a cell. They're out of proportion, right? Am I making some sense? Okay, let's. What is flow of energy in living organisms? Energy flows into an ecosystem as sunlight and leaves as heat. Okay, photosynthesis generates O2, carbon generates oxygen and organic molecules which are used in cellular respiration. Cells use chemical energy stored in organic molecules to generate ATP, which powers the work. This chapter, I do not have much writing in this chapter. I have a lot of pictures in this chapter, so stick with me. Try to stay with me. Do not drift away and fall asleep or daydream. Evelyn, yes, I'm going to say Evelyn, I mean everybody. Evelyn is paying attention 100%. She should be a role model for the rest of us. Okay, so, but anyhow, you do have chloroplast right here, which absorbs sunlight, and then chloroplast, with help of oxygen, breaks down the food, and mitochondria, of course, breaks down the food into ATP and carbon dioxide and water. Carbon dioxide and water used by chloroplast, and chloroplast gives off oxygen as its product. You will learn this, what happens, gives off oxygen, chloroplast, and oxygen is being used by mitochondria, and mitochondria will give off carbon dioxide and water. So this is the flow of energy, and this is what God or Mother Nature, or whatever you want to say it, designed for us. The bottom line is here. On planet Earth, President Obama, you and I, and who else? I don't know. Brad Pitt, is that right? You heard about him recently. Brad Pitt, we all go through this. Okay. Brad Pitt, before, I guess, I will not say anything negative anyway about it. It's none of Brad Pitt's business if I say something bad. Goes through this. He uses the product that's photosynthesis put out. He eats it. He becomes strong, and he goes in the movies. He becomes gladiator. No, that was not Brad Pitt. It was somebody else. Anyhow, he becomes what? Alexander the Great. Here we go. Brad Pitt played Alexander the Great, right? Have you seen the movie? No, don't worry. It was not.
He played Alexander the Great?
Yeah, he played Alexander the Great a few years back. A lot of years back. Not a lot of years back, but I saw the movie. Gladiator. Troy, yes, Troy, thank you. I thought it was Alexander. But who played Alexander the Great?
I don't know.
Yeah, it was him too. I think it was Brad Pitt. Troy, yes, he played Troy too. Troy, Alexander the Great. He did a horrible job in 12 Years Slave. You've heard that movie?
I've never seen that.
I thought it was not good. I'm sorry.
I heard it was good.
I saw the movie, and
Bad?
it was good, but it was all hyped up that it was good. It was okay. It's not that great, I think. Maybe because Brad Pitt was in it. He made it actually. Did you guys know? 12 Years Slaves, he made it. He made the movie, he produced it, he directed, he did everything for the movie. But anyhow, what are the catabolic pathways yield energy by oxidizing organic fuels? Catabolic pathways release stored energy by breaking down complex molecules. That's catabolism. Last time I wrote it on the board for you guys. What does catabolism means? What does catabolism means? Anybody? Catabolism means what?
To break down.
Metabolism is catabolism and what?
Anabolism.
And anabolism. Have you heard of anabolic steroids? What do anabolic steroids do?
They build.
Build up, right? That's what steroids do. They call them anabolic steroids. They don't call them catabolic steroids. They call them anabolic steroids to build up, right? Anabolism, build up. And catabolism, what? Break down. Together they make up what? Metabolism. Together they make metabolism. So the breakdown of organic molecules to is exergonic. So when you're taking a sugar molecule and you break down your cells, right now is doing that. Right now as we speak, your cells are breaking down the sugar molecule you ate for breakfast this morning or last night. You might not have breakfast, but I'm hoping you had dinner last night. You didn't. If you didn't have dinner last night, yesterday lunch. Should I go farther back? Yesterday breakfast. Oh, that's enough. Okay, that's enough. Yesterday lunch. Whatever you had, right now your body is breaking down those sugar molecules. Okay. Fermentation is a partial degradation, partial breakdown of sugar molecules that occur without O2. You all know that. When at home you make wine, you make beer. If there is O2 involved, your wine and beer becomes vinegar. Right? Are we making it up or you know that? You already make wine and beer at home? No, I don't either. I've heard that people do that. Okay, so that's fermentation. Aerobic respiration occurs, organic molecules and O2 do you guys watch hockey final? And the final team that wins the trophy, they hold it up like this. I'm holding up the hockey trophy. Stanley Cup. Thank you. I'm glad this thing came off. If it came off, then I would be in trouble. Stanley Cup. This is it. Mitochondria right here. Aerobic respiration. Aerobic respiration means with air, with oxygen. Wine without oxygen. Beer without oxygen. Yes? No? Brian, we didn't pick on you today. I'm sorry about that. You make wine at home? Beer? No? You just drink it. You don't make it. Yeah, we're laughing to get it. We're not laughing at anybody. Anaerobic respiration is similar to aerobic respiration, but consumes components other than oxygen, and we'll talk about that. How catabolic pathways and production of ATP are related. The breakdown of organic molecules is exergonic. I said that. Exergonic, it means it releases energy. When a molecule breaks down, exergonic releases energy. Endogonic, it means it stores energy. The chemical reaction that stores energy is called endogonic. A chemical reaction that releases energy is exergonic. Okay, so fermentation is partial breakdown. Didn't I go over this? I thought I went over that. No? Anaerobic respiration is similar to aerobic respiration, but consumes compounds other than oxygen. I thought I went. Cellular respiration includes both aerobic and anaerobic respiration. Is often used to refer to as aerobic respiration. Although carbohydrates, fats, and proteins are all consumed as fuels, it is helpful to trace cellular respiration with the sugar glucose molecule first. So this is a true statement. This is something you are going to see at the end of this chapter, that you will see the steak you had the egg. Oh my God, the egg you had this morning. The egg you had this morning had protein, right? And the oil you fried it with, scrambled it with, those also will turn into energy. Those will also undergo cellular respiration. And you will see that at the end of semester. But at the end of this chapter, not semester, at the end of this chapter, but it is easier to understand the material if we first go through carbohydrates, go through glucose. Am I making some sense, everybody? We will get into fat and proteins, how they turn into energy, to ATP. But first, let's focus on this one. Let's focus on the cereal or pancake you had this morning. Okay, carbohydrates. C6, what is this one? Refresh my memory. C6H12O6. Oh, what is it? Oh. That's what? Anybody? Tron? What is it? C6H12O6. Anybody? I'm picking on throwing at me. When I say throwing, it means everybody. It's what?
Glucose?
It's a glucose molecule. It's a glucose molecule. C6H12O6 plus oxygen gives you carbon dioxide. That's what you're exhaling right now. Right? You're exhaling carbon dioxide, and of course you give off some water, and of course energy. Without having breakfast this morning, you would not be able to sit down, for example, or you will sit down here and listen to me. You do not have enough ATP. Without breakfast this morning, I would not be able to stand before you and talk. Ask me, Amir, don't eat for a week, and then come up here and lecture. I say, forget it. Not in my lifetime. So I have to. Anyhow. What is redox reaction, oxidation and reduction? Oil rate, oxidation loses and reduction gains electrons, and as this goes on, I guess I will elaborate on it more. The transfer of electrons during chemical reactions releases energy stored in organic molecules. This released energy is ultimately used to synthesize ATP. Again, back to the principle of redox. Chemical reactions that transfer electrons between reactants are called oxidation reduction reactions or redox reactions. In oxidation, a substance loses electrons. So that's what oil, oxidation loses electrons. Am I making some sense, everybody? Why did I pick up on oil rate? And then rate, reduction, it means what? Gain. Opposite, they're kind of reduction. I mean, I thought reduction means reduce. No, not in here. In biology, everything is opposite. The mentality of biologists are cuckoo. So oxidation, when the electron is being gained. Okay, I hope it says it somewhere here. In oxidation, a substance loses electrons, is oxidized. And in reduction, a substance gains electrons or it is reduced. When it gains electron, it is reduced. The amount of positive charge is reduced. That's what the reduced means. The amount of positive charge is being reduced on a molecule. Here they are, an example of it. Amir, what is an example of it? Sodium and chlorine. Okay, so sodium in this case becomes sodium. You remember from beginning of semester? If you remember this from beginning of semester, they had 2 of them here, 8 of them here. Is that right? You remember that? 1, 2, 3, 4, 5, 6, 7, 8. And the last one had how many? 1. Sodium. Okay, and chlorine from beginning of semester he had 1, 2, 3, 4, 5, 6, 7, 8. I think the third shell had 7. 1, 2, 3, 4, 5, 6, 7. Right? Sodium gave this one to that one. You remember that from beginning of semester? And sodium became what? Positive. And chlorine, since it got 1, it was negative, right? Because imagine if I get money from you. You are sodium, you give me $1. I am in whole $1. Right? I am in whole $1. And then of course you're $1 ahead because you gave me $1. You're not in whole. I am in whole. Y'all, those terminologies I'm using, y'all know what I'm talking about? So that's why chlorine is negatively charged, because sodium transfers its electron to it. So that's what we're saying here. Sodium becomes oxidized because it gives off its electron. And chlorine is reduced. It is in whole. It got an electron from sodium. You remember this from the beginning of semester. So sodium, it is oxidized and chlorine is being reduced. It gains electrons. From where? It gains electron from where? From sodium. Remember this from beginning of semester? Okay, so I did explain oxidation reduction a little bit. Well, not a little bit, very good. I have to, what is it? Play my own drum or blow my horn, something like it. Brian, what is the right terminology? I have to blow my horn. Yeah, it's too early in the morning. Reducing, so this is giving a reducing agent would be the Xe and becomes oxidized, and then becoming reduced. Of course, the Y is becoming oxidizing agent. He's giving you all of that electron that I talked about. It electron. Sodium and chlorine would be equivalent to that. The electron donor is called the reducing agent. Electron donor. Sodium is electron donor, so it is reducing agent. And the electron receptor, receiving electron, is called oxidizing agent. Do I make sense? So chlorine is oxidizing agent. Can you guys see that laser pointer from back there? Stephen? Yeah. You can see the pointer, laser pointer? Yeah. Right here. So if you forget these at home, go draw this for yourself one more time. Maybe I should do this. That makes sense. This one electron from here goes to here. Now this one has 8 electrons, and this one, this vanished, this disappeared. Outside has 8 electrons. Go back to your exam number 1 material. Look at that chart. If you still, I did draw it for you, but yeah. During cellular respiration, now that was sodium and chlorine, but during cellular respiration, the fuel such as glucose is oxidized and oxygen is reduced. Glucose is oxidized and oxygen is being reduced. Okay? So here they are. Oh, I lost it. I guess the pictures are not here for whatever reason. I probably copy and pasted from another place. So glucose is oxidized and oxygen is being reduced. just like that chart that I said. Okay, what is the stepwise energy harvest via NAD and electron transport chain in cellular respiration? Glucose and other organic molecules are broken down in series of steps. Electrons from organic compounds are usually first transferred to NAD, a coenzyme. We talked about that. And as an electron acceptor, NAD functions as an oxidizing agent during cellular respiration. Each NAD, all of this will make sense with pictures. If I read it to you, it's just, I hope, here it is. Okay, great. So this is the picture of NAD. Let's take a 5 minutes break so you can eat your banana, apples, whatever.